So yeah, I'm going to show you guys bed format. So what is the bed format? Well, it actually stands for browser extendable data, which is kind of interesting. I never knew that until today. Basically, if you experience a file format in bioinformatics that you're not really familiar with, there's typically a Wikipedia page on it, and that's how I figured that out. Quite simply, it's a file that contains genomic regions. And what's unique about it is that it uses zero-base positions, and I think probably many of you are familiar with zero-base positions, but we'll go over it if you're not. Typically, they're tab delimited, so just pretend that they're always tab delimited. They can technically be space delimited, I believe. And I highly recommend getting familiar with the Bedtools command line interface because it's extremely powerful and very fast. And so here I just have an example of a simple bed file. You can see that for chromosome 1, it does start with the number 0. And there's a reason for that. It makes certain problem solving with computation much easier and much faster. Okay. So it, at the minimum, just requires a chromosome, a start, and an end position. So, but there are two kind of flavors of bed files. The first is what I assume is what it was created for, was for the UCSC Genome browser. And basically, the other flavor is everything else that you can use with it. So for the UCSC Genome browser, it essentially will require all this information, but it can still operate with just the first 3. However, if you start putting entries in the 4th, the 5th, the 6th column, and so on, it will expect it to be like this. You don't really have to worry yourself too much about it. Making a bed file for the UCSC Genome browser is very good for visualization and also for like making figures and stuff. I actually have a very quick tutorial on that at the very, very end. So it's just something to keep in mind. Now, the real thing about it is that when you use it for everything else, you can append more information to a genomic position, right? So you can essentially tag information or anything you want, like gene names, or maybe like a certain score for a gene, or a certain score for a variant, or genotypes, or anything really, to a position. And once you have it in this format, you can use the Bedtools suite in order to intersect things, filter things, and just kind of like perform bioinformatics in a very fast and efficient way, which we'll go over. So now let's talk about the difference between the 0 base and 1 base. Quite simply, basically you start numbering the chromosomes at position 0 and not position 1. Now, if you want to convert from one system to the other system, you simply just add 1 from a 1 base position to convert to a 0 base, and the end position stays the same. So let's work on an example of this. Okay, so I'm going to annotate this, and now we're going to look at the single nucleotide here. So in 1 base position, it's saying chromosome 1, position 4 to 4. I mean, that's quite simply just this. So it's a G, right? But in 0 base, you're kind of looking at the numbers in between the nucleotides, right? So if you wanted to get the G, you have to include both 3 and 4. Hopefully this makes sense. It's not too much of a stretch, I hope. Now let's look at one where you're looking at a range of nucleotides, which is something that you see with like structural variants or indels or other things, right? So here it's going from 2 to 4, right? So 2, 3, 4, right? ACG. That makes sense. Now, to get to the same position or the same nucleotides in the 0 base coordinate system, it's going to be 1, 2, 3, 4. Now, hopefully you can appreciate by looking at these two examples, converting from 0 base to 1 base, or vice versa, is simply just leaving the end position the same, so you don't touch it, and only manipulating the start position. So, the rule of thumb is basically just you subtract 1 from the 1 base position to get to a 0 base, or vice versa, you add 1 from the 0 base to get to the 1 base. So, why do we have 2 coordinate systems in biology? That's a really good question. I don't know, but you can use this guide in order to help. Right, let me stop drawing. So basically, this is also on the Gleason GitHub page, and I can share this table too. It's actually on the homepage of the Gleason GitHub page. So bed files are 0 base. Now all these other file formats, some of them personally I'm not that familiar with, like Wiggle. I've heard of it, but I don't really work with them. Will tell you if they use 0 or 1 base positions. A kind of bad rule of thumb is that if you can read the file, like if it's human readable, it's typically in 1 base position. Exceptions are bed files, because you can read them and they're in 0 base positions. This applies, for example, with BAM files. BAM files are a binary file that you cannot read if you try to look at it, right? So they use the 0 base position. If you use SAM tools to view the BAM file, you're now converting the binary to a human readable format. So now you can read it, and now it's in 1 base position. So this is just kind of like a nice, handy, guide in order to kind of, know what you're looking at. So now let's actually do the first exercise, which is let's convert a VCF file to a bed file, and we can use awk in order to do this. Quite simply, we're just going to print SNPs into a bed file, first by grepping things that are not in the header by using this V operator to grep lines that do not have this pound sign. And this Omni file is basically it's a microarray of 1,000 genomes samples, and it's used in the GATK best practices. Like if you wanted to call variants and stuff, you're probably familiar with this. Now the trick of it is like what we mentioned. The VCF is a 1 base file. The bed file is a 0 base. So now you're just going to subtract 1 from the start position in order to print the variants. So let's do this. Okay. So in this directory, I have a lot of stuff because I already ran it. But quite simply, what we're going to do is I'm just going to copy and paste it, the command. So we're going to run this command, right? Now here I have other information, which is the benefit of making bed files, right? I have the position. I'm still going to print the SNP, the SNP name, the referenced allele, and then the alternate allele. And it shouldn't take too long to run. And then we'll take a look at it. Okay. So as you can see, it's like I said, chromosome start and SNP reference alt. And for a single nucleotide, it is you can tell that the numbers are not the same, right? But if you subtract the n from the start, you get 1, which is the length of a single nucleotide variant. There should be possibly some indels in here, because I think there are. But if not, they would be longer than 1. Okay. Now, if you look at the size of this, it's quite big, right? 50 megabytes, right? So, so in order to compress it, you can use bgzip and tabix. Now, before you do this, you need to sort your bed file. So, yes. So the reason why, like I said, you can intersect it to regions. Bedtools can take a VCF input, but if you have indels or if you have SVs that have an end position somewhere in the VCF, it won't consider that. So this is a really good reason to use a bed file if you have a VCF of structural variants or something else. Okay, so as I said, Bedtools really likes sorted files. The reason why is that it can actually run a lot faster if you use the sorted flag, which I'll give you an example later. I typically like to use the Unix sort command, because you can use other options like a temporary directory for very large files. And so that's what we're going to do right now. Also, before I go any further, you'll probably notice how I started naming the files. And this is just good. It's good practice if you plan to share data, right? You know, if you're working with yourselves, you know, maybe you don't really need to follow these rules. But, you know, if you're planning to share the data to other labs, to other people, it's good to always kind of explicitly say in the file name the genome reference that it belongs to. Okay, so now let's sort it. So we're going to do sort. Basically, what this says is to first, wait, let me get my mouse, is to first sort it on the chromosome and then sort it numerically on the start position. And I personally like to do this what I call like in-place sort. So I just, you know, make a temp file, put a semicolon to end this command, and then I just move the temporary file to the new file. And so now it's going to kind of, it's not really sorting it in place, but functionally it is. Okay, so now it's sorted. Now we're going to compress it with bgzip. Now I've already done this, so I'm going to use the F flag to force it. And it shouldn't take too long, and it'll compress it. And now if you look at the size, it's going to be much smaller. And it's going to be, it's going to have a .gz extension. So you can see it basically cut it in half. Now if you tabix it, you want to specify that it's a bed file, and you do that with this pbed. The reason why you want to do this is that it can make, you can use the tabix command line operator in order to subset the file and to do other things with it too. But we won't really talk about that nowadays, or right now. Okay. So now after we've done that, you know, that's basically, you know, how you can, you know, convert a VCF file to a bed file, sort it, and then compress it in order to make it a lot smaller, so you can save up on disk space. Yes. Okay, now we're going to use awk. Now this is kind of a little tangent. I don't really do this too many times, but sometimes you'll have a bed file which is very large, and for whatever reason, you can't really run some command on the whole thing. So maybe you want to split it up by chromosome. A good way of doing this is with awk. So the basic idea is that you're going to be using this append operator. Now this doesn't work with the regular output operator, because you're essentially going to be writing over every line. You're going to be writing over it, right? You can test it out if you want. But so this works with the append operator. However, if you make some sort of mistake, I mean, you know, I do this a lot. You know, I make mistakes a lot. Sometimes you need to delete the files and run them again. If you do this, or you know, if you need to run the command again, delete the files first, because it will just append to whatever you've done before. So you know, actually, I've done this before. So let's actually delete these files. So I think if I just do issue19 bad, I don't want to delete all of them. So I'm just going to do omni. Okay, I think, yeah, those I can delete. I'm just going to delete them. And now I'm going to do a zcat because we compressed the file. Issue19 awk. And so here what I'm saying print the whole line. This $0 is the variable for the whole line. Append to the, and here we're just giving a prefix, omni 2.5 period chromosome name. So here it's going to append lines that have chromosome 1 to a file that has chromosome 1 in the file name. And then as a string, bed. And that's it. You know, it almost looks like the command. It almost looks like that, like it wouldn't really do this, right? Like when I first learned about this, I was like kind of skeptical because it's like, really? Like, I don't know. but it actually does it. And you can look at it by counting the lines, and you can see that, you know, especially if you sort it, reverse sort numerically, you know, now you can see that, you know, chromosome 2 has the most, and then chromosome 1 does. You know, actually, I think in this reference build, chromosome 2 might be a touch longer than chromosome 1. We'll come to this later. Sorry, that's spoiling it. But yeah, you can see how, you know, it really did split it up. And, you know, if you want to look at the chromosome X one, you'll see that these variants are all in chromosome X. So that's a nice little, you know, nice little thing to learn. Okay, so now I want to talk about using Bedtools specifically. So you made your bed files, now you want to, you know, do some bioinformatics on it. So a lot of Bedtools has a lot of commands. You know, you can just type in Bedtools in the command line by itself, and then you can see everything that it contains. This genome arithmetic, here I'm using version 2.27. I believe it's 2.29 currently. But in either case, you can see that a lot of these commands in the genome arithmetic are actually really informative for doing very specific operations. I've used probably about half of these at one time or another. And I would probably say that honestly, about a lot of the times when I first learned how to use it, I was using some other script that I wrote before. And I kind of wish I read the documentation a little more carefully in order to, you know, like make my life easier. So yeah, definitely, you know, search online, you know, a subcommand that you might think might help you, or, you know, you're not entirely sure. There are really good visual examples on it. And here I'm showing you the intersect command, which is like kind of the bread and butter of Bedtools. As you can imagine, the intersect command is basically asking, of the regions I have in the A file, which one of them intersects, overlaps with the ones in B? And there are a lot of options that do this. We'll actually use this V option for filtering variants because it's similar to that grep V operator, where it looks at the variants that do not overlap the stuff in B. So it's very useful. There's also documentations for most of the options for a subcommand. So here's one that I've used quite a bit in the past, is the WAO, which essentially just writes the, as it says, the amount of the overlaps. And then, you know, you can look at the example to see that, you know, to understand how it works better. Okay, so now we're going to show you a practical example of using this intersect command, and we're going to filter variants in bad regions. So this is the basics, you know, structure, and this V option is what I mentioned before. It's similar to the grep one. Basically, we're going to do the opposite of what we tell Bedtools to do. Okay, so I have this file called segdup HD19. Oops. And in it is basically just regions of the genome that are very noisy. Essentially what they are just rather large repeats that are scattered out throughout the genome, and homologs of segdups are about 90% identical. Now, the reason why you may want to filter variants that are in this region is that with short read technology, and even other genotyping platforms, it becomes difficult in order to get like a good alignment or a good call in that region. And the reason is because it's repetitive. So many times you may want to filter your variants if they intersect these regions, because you're not really confident of whether or not the genotypes are in fact valid. However, to do this before, I recommend doing a couple things. So first is sorting the file. And I'm going to do this kind of thing where you know, I'm just like doing this like in place. Now you may ask where I got this file. I got it from the UCSC Genome browser, and maybe later on I can show you an example of how to download data from the UCSC Genome browser. Okay, now the reason why we're going to do this next command may not be that intuitive, so I'm gonna draw it out. So sometimes we have a situation where you have a variant that say is rather large, like this, like a deletion or something, right? And now you have your seg dupes, and say I have a seg dupe here, a seg dupe here, maybe one like this, because there are some overlapping positions there, and you know, maybe one like this, right? And maybe make this a little bit longer, just for the sake of argument. Okay, so I want to run an intersect command asking for the variants in A, remove them if the total overlap to B, or the seg dupe file, is 50% or greater, right? Now we can look at this guy, and this is probably like maybe you know, 30% or something, or not 30%. It's like 3%. Sorry. Now these in total may be about, you know, half the length, but the problem is that they're broken up, and that individually this maybe is 5%, and maybe this is 25%, and this is, you know, maybe like 30% or something, right? But they're broken up, and so if we use the restriction, what I'm going to show you, the f 0.5 option, it's not going to work. So to overcome that, what we're going to do is we're going to merge, and essentially when you do the merge operation, it's now going to merge these guys into one unit. Granted, this still does not complete the full expectation, which is you need to also count this overlap with this overlap, and you can do this with a subsequent script. I'll actually give you an example of how, you know, you can couple Bedtools output with writing a script. And so perhaps if you wanted to solve this problem by yourself, you could do it writing your own script. But I'll probably give like hopefully a good example of that later. Okay, so to merge this file is quite simple. You have to sort it first. Merge bed, I for the input, and then just give it the file name, and then I like to write it like this: merge. Yeah. Okay, and that was it. And now if we look at it, actually, you know, if we just count the lines, you can see that there's quite fewer lines in this because we've merged it together. Okay, so now let's get to actually filtering these guys. Okay, so you do intersect bed. You give the A file. So here we're doing 1000 genomes omni HG19 bed. And Bedtools can take the .gz or the .bed. It can work with both. And now we're going to give the seg dupes merged. We can give the sorted operator because it'll run a little faster. I mean, this runs pretty quickly. We're going to say write the entry in A, because by default, Bedtools, and you know, you can look at the example online, will report the positions that overlap. So if you want the original position, you need to define write A. We're going to give this 50% overlap requirement, and now we're going to give this V, which is now going to basically do the opposite, right? So now it's going to say, give me the variants that do not overlap 50% or greater. And now we're just going to write it out. Filtered. We'll get there later. Bed. And it should be pretty quick. And then we can count the differences. So first I'm going to do zcat because the file is compressed. Let's do a word count. And you can see we have that many. And for this, yeah, so, it removed, quite a bit of stuff. Yeah, so like 40,000 variants, yeah. So, that's like a pretty, hopefully a good practical example of filtering variants using Bedtools. And that, I mean, there I did it right, right for you in like less than 5-10 minutes. Okay, let's see what else we can do. Oh, okay, now you want to make a bed file that contains exons. This is a little bit more involved, so I'll walk you through it. Zcat gencode GTF. So GTF was that file that, if you remember a few slides back, right here, it's one based. So that's something that always keep in mind. Typically, it's a good, that's a good thing that, like, when you're given a file, and you look at the extension and you're not really sure about the position, yeah, try to look it up or refer to that table. And then, over time, you'll just know, like, oh yeah, GTFs are one-based positions. So first we're going to, open it up. We're going to grep the protein coding regions, or the protein coding transcripts, because there are transcripts that are non-coding that are also in these GTF files. Essentially, a GTF file, if I didn't mention, it contains transcripts. Also contains exon positions. And so we're going to take the third column, which is equal to an exon. Then we're going to convert the one base position to a zero base, and then print the entire line. The reason why is that for me, when I tested this out, awk would try to split also on the white space. And this will be more evident once we look at the file. So in order to overcome this, I know that cut will respect the tab delimited files. And so here I'm just taking the positions that we made and then the last column that has the information. So let's look at this file. And if you're interested, this file is on storage. So I can point you to it later. So you can see it has, let's get my mouse on that. You can see it has a gene, which is the entire gene body, the transcript, which includes just the parts that RNA Pol II produces, and then like the exons, right? And there's some other things, like you'll see that for coding regions, you'll have the position of the start codon and the CDS, which is the actual region of the gene that's for coding, and UTRs and stuff. So this is essentially what we're doing, right? Is we're doing chromosome matching on exon. There's your one base position, and then here's your information, right? And so you can see right here is the gene name is a SAMD11. Okay. So I'm just going to paste this command because it is quite long. And if you're interested in how I came up with this command, it was just, just trial and error, But overall, you can do this in the command line. And so yeah, as it's going, if you run a Bedtools Intersect with the Omni file that we just made, and this GTF file, you'll get this warning. And this is pretty common. It will say that the inconsistent naming convention. Now, if you get this warning, this typically means that you have one file that has a CHR in it and one that doesn't. So another practical example is how to fix this. I typically use Perl one-liners. That's because I'm more familiar with it. I know you can do this in SED, but I don't know SED. So this is how I do it. So if you wanted to add CHR to the beginning of each line, you can run this command in the command line. But what we're going to do is we're going to remove it. So you basically just flip it. So if you're not familiar with regular expressions, this caret just means the beginning of the line. And so here, what this is saying, it's saying, Oh, wait a minute. No, sorry. Yeah, this is removing. This is removing CHR in the beginning of the line. I'll fix this. Sorry. And this is adding CHR to the beginning of the line. I flipped it. So this, what this is saying, it's saying match the beginning of the line CHR and then replace it with nothing. And the G just means do this for every line. Do this for every instance you see of that. So let's do that because we're going to get this error anyways. Because if you didn't catch it, it has that CHR in the beginning. So it's a simple Perl pi. That's the way I remember it. Is like, you know, PIE S for beginning of the line CHR with nothing G, and then you can give the name of the file. But this is actually really powerful. So if you know, say for example, if you wanted to add the chromosome to the beginning of a lot of other files, you know, you could do it like this. Here I'm flipping it, but now I'm going to do it for those chromosome files, and you can use like wildcard operators in order to achieve this. Okay, so now if we look at it, we removed it. So now we shouldn't have this problem. But if you're interested, this is quite large. So I already sorted it. So let's do the bgzip and the tabix command. Oh, that's not gonna work. Forgot to put bed, and I think I have to force it. So yeah, we'll let that run. And then after that, we are just going to intersect it using Bedtools. So now let's intersect it. Intersect bed. So now, so the A file is the one that is kind of like your reference, right? And so here it's the variance. So omni filtered bed, or HC19 bed. B, now we're going to give the gen code exons, and it is sorted. So we're going to say it's sorted because if you don't give it, it will actually take a lot longer to run. And now we're going to do write A and write B. So now it's going to give us both the entry in A and the entry in B. And now I'm just going to write it out to the filtered exonic HC19. And it shouldn't take too long to run, actually, because we gave it the sorted flag. Okay. So yeah, that's basically, you know, the same thing. And now let's, you know, now like what we're going to have is for some variants, and especially for variants that are much larger, like structural variants or other things, you may intersect more than one gene. So what if we wanted a file where it's the variant and a list of all the unique genes that it intersects to, right? And it's like a comma separated list. Now the way that I would do this, I'm sure there's other ways to do it, is to use a dictionary of dictionaries. So that the first key is the variant and the second key is the gene name. And by doing it this way, there are some advantages. You can count the number of exons per gene, which I'll show you in an example right now. And you can also, and then you'll be sure that you're looking at unique things and you're not like duplicating stuff. So yeah, now it's done. Now let's take a look at it. So you can see, so yeah, you can see right here that you have one variant or one SNP that's intersecting different exons, right? Or different transcripts of the same gene. And so basically we just want to record the unique gene name. Hopefully, yeah, maybe this one doesn't have it, but other ones will. So let's look at the script. So the script's also in here. I'll put it up on GitHub too. And it's called report all exon overlap, and I wrote it in Python. It's a little long, but you know, I'll quickly walk you through it. So here I'm using default dicts because I find that this will be a lot easier if you use a default dict. The basic idea is to create a data structure where a variant can point to all the genes it overlaps to. We can use a dictionary of dictionaries in order to do this, right? So you have a dictionary of the variant name. This points to another dictionary that has the gene name and the number of times, the number of exons that the variant has overlapped to that gene. Okay, hopefully this will be a little bit more clear. It might be a little confusing. This would be more useful for larger variants, right? Where you have like something that spans like multiple genes. And so, you so that you'll know like the number of exons that will hit. Okay. So here I'm just, here I define the dictionary of dictionaries. And I'm opening up the file that we just made. And I created the key of the variant, which is just a simple tab delimited string of just chromosome start and ref, SNP, ref, and alt. And just to make sure I did it right, I typically just print it to the screen, after I did it, and just just make sure that it's good. And this is just good practice. I mean, probably it's a lazy way of debugging it, but it works. So in order to parse the gene info, maybe this is a little bit overcomplicated, but to me, I thought it was easier if we just made another dictionary that had all that information. So, first we can remove the double quotes, and I'm not going to really walk you through this kind of massive code, but essentially sometimes there were 3 elements in a string because of a beginning of there was an empty character essentially. But basically now we can skip lines that do not have a gene name, right? And now we can get the gene name through a dictionary. And I printed it out just to make sure that it works, and it does. Okay, so here is where we're kind of like loading the dictionary, right? So I'm saying if the variant and the gene name has never been seen before, if you get the value and you don't find anything, then just make it equal to 1. And if you see it, and this is the kind of benefit of the default dicts, is that I didn't have to do this for the gene name. Then you just add 1 to the value. Okay, so here now we're going over everything. So now I can get the genes by looking at the keys. You can join them by commas. And if you wanted to do the counts, you could do something like this, right? And now we're just going to print it out. This is quite a large file that we've created. If you look at it, 37, right? So it does take a little bit to run. So yeah, we'll just give it some time. All right. Well, hopefully that was clear. And then while it's running, the last thing I'll show you is how to put something on the UCSC Genome browser. Now, the reason why that's nice is that sometimes you just want to do like sanity checks, and for me, working in bioinformatics, you tend to just get used to like data, right, on as text and things like that, and you might be a little detached from the biology of it, right? So by putting your data into the UCSC Genome browser is a good way to kind of be like, yes, these are variants that are hitting genes, and it's real biology and stuff, like these are real people that have these variants, and so it's a good like frame of reference to do this. Okay, so now if you look at this file that we just created, you can see that this is what we have. And for example, some of these entries intersect more than 1 gene. So that's kind of cool, right? Right there, we just annotated some variants in like 45 minutes, right? All right, so the very last thing is to make the UCSC Genome browser file. So basically, you can just run this command. We like look at it, and we can cut. I guess I could just cut 1 through 4. Now, the thing about the UCSC Genome browser is that they really like to have the CHR prefix. Like it won't work unless you do it. So we're going to use this Perl command in order to add the CHR prefix to it. It's really quick. And now let's look at it. And now you can see that this is, these are the file that we're going to upload to the Genome browser. Okay, so I actually already have this. I already have this ready. on my local computer. So if you go to the Genome browser, you go to My Data, Custom Tracks. You may need to make an account. It's free. Always make sure that you're working in the organism that you're working in, and also the genome build. So here we're doing issue 19. That's why I said that if you're sharing data with other people, it's always good to mention it, like in the file name especially, so that there's no questions about it. Okay, so now we're going to upload the file, and it's very quick. And then you can read about the format, how to like put colors and names and all these things to it. It's really, you can get really intricate with it. So right there, you can see your user supply track. I prefer like looking at it with pack. And so you can now see the SNP name. And, if we, go out quite a bit, let's go out even more. Hopefully you should be able to see some SNPs, and there they are. And those are the ones that intersect the genes. And, if you want, you can just kind of explore it on yourself and zoom in and, all this stuff, right? You can have fun with it. So that's basically it.